This chapter is one of 3 chapters. We're going to talk about energy. And energy is a really important part of life. Without it, I can't talk to you. You can get up and move around in the morning or do really anything. And so what is energy and how does it apply to biological systems and life? So energy is the ability to do work, and work just means being able to kind of move or transfer molecules from one place to the next. And energy comes in many different forms. You might have heard of kinetic energy, which is energy as it relates to the motion of different molecules or things. Potential energy, which is energy that is stored up and that could be used later. And thermal energy, which has to do with heat. Mechanical energy, that has to do with movement of joints and things of that nature. And this class, we're going to focus a lot on chemical energy. And chemical energy is energy that is stored up, or potential energy that's stored in bonds. And so when we talk about chemical reactions as it relates to how our body works, there are a lot of different molecules that are forming bonds and breaking bonds. And so how is energy used or stored in relation to those processes? So that's what we're going to talk about a lot. All right, so something that you should understand about energy in general are the 2 laws of thermodynamics. And the first law says that energy cannot be created or destroyed. So energy can be transferred from one form to another, but it's not going to be necessarily lost and disappear once you used it. The second law of thermodynamics says that when energy is transferred from one form to the other, sometimes some of it can be released in the environment as heat. And this is important for us to understand, because basically this means that our energy, and as it's released and moves from one form to the other, is going somewhere. And where does it go, or where does it come from? And to understand this concept, you have to understand a little bit of how molecules are organized and what's the relationship between that and the stored potential energy. So I want you to think of it this way. So let's say you have a molecule. So each of these dots represent, let's say, a carbon atom. And they're connected together. This line means it's bonded or connected together. And so molecules are usually found, with bonds holding them together. But chemical reactions can break these molecules apart. Example of that is when you eat food. When you eat food, your food, when it's used by itself, doesn't go in as like an entire, grain of rice, or an entire corn kernel, or an entire slice of bread. It's actually broken down into pieces and smaller molecules, and even smaller than that, for your body to use it and convert it to energy. And so in order to do that, there are different types of reactions that can take molecules from big form to small form. And vice versa, if our body is producing something, it can build it up from small pieces into one single bigger molecule. And so molecules and chemicals can be found in pieces. So this would be one piece where you have 2 connected here and a separate piece over here. Or it could be joined together with 2 bonds between these 3 atoms, okay, in my little example. Now, in between these 2 different forms, I'm showing that you can go back and forth. So you can either go from having them separated and join them together, or you can have them joined together by bonds and break it down to separated pieces, okay? So there are terms for this, and that's what I want to teach you now. The more separated a molecule is, or the more pieces of molecules that are floating around or existing in the environment, we have what's called entropy, higher entropy. And entropy is the measure of disorder within a system. So if you had a molecule that had originally 10 bonds holding it together, and you broke 3 of those bonds, I think you'll end up with about 4 pieces now of that molecule. Some bonds would still be there, but it would be broken into pieces. And once things are separated, they can spread out, they could interact with other things, it becomes more disordered. and that means you have higher entropy. But once the molecule is stuck together and all those bonds are holding it together, it's more orderly, you have less entropy. So when it comes to talking about energy, we want to first understand this concept of how molecules are associated together, or how they can be disorderly and disconstructed. Okay? Now, when it comes to the molecule actually being held together by bonds, the more bonds that are present within a molecule, the higher the potential energy that molecule has. And potential energy, just think of it as the possibility for something to happen. And when you think of bonds, think of them as the possibility to break a bond. Okay, so how many possibilities do you have to break a bond if you have a molecule with 10 bonds versus a molecule with 5 bonds? The molecule with 10 bonds have higher possibilities or potential for those bonds to get broken than a molecule with 5 bonds. Okay, so the technical term for potential energy stored in bonds is enthalpy. Okay. So in these two examples here, this bottom one has higher enthalpy than the top one, okay? Because this one has 2 bonds, this one only has 1 bond. So now we have to connect these two things together. We just talked about entropy, we're going to connect it with enthalpy. So when you have more bonds, you have higher bond energy stored, potential bond energy, or higher enthalpy. However, when you compare this molecule to this one, this molecule is more organized or more orderly than this one, okay? So this molecule would have lower entropy. So it's more organized, therefore it is not disorderly, okay? The reverse is that up here, this molecule has less enthalpy than this one because it has less bonds, less potential to break those bonds. Therefore, it has less enthalpy than this bigger molecule. And when you have less enthalpy, that's because your bonds are already broken and you're disorganized. So you have 2 pieces versus 1, and these 2 pieces can go anywhere. That means this has high entropy compared to this one. So it's a relationship in relation to each other when you look at these different concepts. All right, so enthalpy and entropy has an inverse relationship with each other, where if you have high enthalpy or high bond energy, lots of bonds to break, you're going to have low entropy or low disorder, meaning that your bonds are together and they're organized. If you have low enthalpy, means that you don't have a lot of bonds to break, and that's likely because you're already broken them. That means you have high entropy because you're now spread out in disorderly molecule. Okay, so go between these two concepts and make sure you understand them together, because that's how it will make a lot of sense for what we're going to talk about next. So as your bonds break and bonds are formed, energy is released to the environment or used from the environment for this process to occur. And so that energy is called free energy, or Gibbs free energy. And that energy is measured as it relates to the bonds being broken and the bonds being formed. So in short, there's a certain amount of energy needed to be added, and it's characterized as this letter G, or delta G, which is change in energy. There's a certain amount of energy needed to form a bond, and there's a certain amount of energy released when bonds are broken. And this is free energy that is released in the atmosphere. Now, this energy, once it's used to form a bond, that energy becomes stored in the bond. And that's where we have higher enthalpy or higher bond energy. And once the bond breaks and the energy is released, now you have less bond energy because the energy is released to the surroundings. This free energy can be used or understood further in relationship to metabolism. And metabolism are the chemical reactions in your body, and you have reactions that are either building up molecules or breaking them down. The same idea as we just mentioned. And so if a molecule is being broken down, it's called catabolism, or if it's being built up, called anabolism. Okay? Catabolism or catabolism, this is basically what we're showing here. When you have a molecule and you're breaking the bond and it's going to be more disorderly or have higher entropy, that's when you're going to release energy to the environment. So think about when we eat a food and we break down our food, we're releasing energy stored up in the bonds of those foods. So even like if we eat bread or pasta, those have what's called complex to sugar or carbohydrates. As we break those carbohydrates bonds, break, we're releasing energy, releasing energy, okay? And our body can use that energy to drive other chemical reactions. And if we need to build up some molecules. So let's say, in our body, we are constantly making different proteins, and proteins are made from amino acids. And once one amino acid comes together with another one, that needs to form a bond. And that forming a bond needs input of energy. That's an anabolic or process or anabolism happening, okay? So if you're going to form a bond, you're going to need input of energy, and that energy comes from somewhere else. And so when you think about reactions, I want you to think about it as working together. So if you have some reactions over here where the bonds are being broken, energy is being released. So every time you break the bond, energy released. That energy, when it's released, sometimes there's a reaction happening close by that needs energy, an anabolic reaction, and that will take that energy in and form a bond, okay? So this is known as coupled reactions, where sometimes you have two reactions happening simultaneously in the same place. One reaction is catabolic, break bonds, release energy. The other reaction is anabolic, where it needs energy to form bonds. Okay, so they kind of work together with each other. All right. You can think about it as, let's say that, I'm a donor and I have money and I want to just give it away, and I'm walking by someone and they're like, I wish someone would give me some money right now. And so as soon as I'm giving money away, it's like, anybody wants this money? Free energy. Then someone says, I want the money. And they said, oh, thank you. Now they got the money and I gave the money up, and we're both happy people. Okay, so think about it that way too. All right, now when it comes to anabolism and catabolism, these are very closely related to these two types of reactions. So you can almost think of them the same way. Exergonic reactions and endergonic reactions are reactions that are either releasing free energy or using free energy. Okay, so this is just talking about reactions as it relates to free energy. And I put it in the same color so you can kind of remember it. So exergonic reactions are reactions that are releasing free energy as a result. Those types of reaction would be catabolic in nature. Endergonic reactions are reactions that need input of free energy. Those reactions would be anabolic in nature because they are inputting energy to build something together, to store energy, potential energy in the bonds. Okay. Now, when you think about endergonic reaction and anabolism, this is going to be, how do you relate it to these two concepts that we talked about here, entropy and enthalpy? So if you have an anabolic reaction, first of all, it's endergonic because you had to put energy in, and you're putting energy in to build it up, to make more bonds. If you're making more bonds, you're going to become more orderly. If you're more orderly and organized, that means you have a little bit of entropy and a lot of enthalpy. All right? So I want you to practice this. If you're exergonic reaction, that means you're releasing energy. Reactions that do that are catabolic in nature. And if you have, and if you're releasing energy, right, you become more disorderly because you broke bonds. If you broke the bonds, you'd have a little bit of enthalpy and you have more disorder, you have more entropy. Okay, so that's how some of these different terms that we're talking about is related to actual bonds and molecules in your body and how the energy is being used and released. Another important molecule for you to know or understand is adenosine triphosphate, or ATP. And ATP is an important energy molecule because this molecule has 3 phosphates attached to each other, okay, to adenosine. And this phosphate on the end is very weakly attached. So a lot of times whenever this bond is broken, it becomes more now disorganized, right? Because the phosphate is separated from the main molecule. So it releases energy, and that release of energy can go and drive other reactions. So it works with other reactions, or it's coupled to other reactions. So when we talk about your mitochondria, and we'll talk about that more in the next chapter, the mitochondria helps to produce ATP, and ATP helps to allow other reactions to take place, because when this phosphate is broken off of the main ATP molecule, it becomes adenosine diphosphate, which just means 2 phosphates. And once the phosphate was broken off, it's going to release energy to the environment. And that energy, G, gives energy that's going to go and help to, which reaction will it help with? Catabolism or anabolic reactions? It will help with this one, right? Anabolic reactions, because they need input of energy. So it helps to build things together, build them up, build up the molecules. All right. And so in order to make your ATP, so when ATP is broken down, it makes ADP, but you can go ahead and make ATP from ADP and phosphates, okay? And in order to do that, would that be a catabolic or anabolic reaction? That would be anabolic. That means you need input of energy to form that bond again, to go from adenosine diphosphate to adenosine triphosphate. Okay, so that energy, you get that from the food you eat. Okay, so that's why when you eat food, a lot of those molecules, especially glucose, goes to your mitochondria, and it helps to take those ATPs that were broken down to ADP and build them back to ATP again. So your cell could have lots of reactions that need energy take place. Okay, so there are a couple things that I want you to understand to wrap this chapter up, and they're very related to what we just talked about. So redox reactions are reactions that are basically working together with each other, just like how we talked about coupled reactions. But these reactions involved loss or gain of electrons as they're transferred from one molecule to the next. And reduction is, so this is a word joined of reduction and oxidation. Reduction is the term that refers to the molecule that's gaining electrons, and oxidation is referring to the molecule that is losing electrons. And so there's a little way to remember it. If you say oil rig, oxidation is loss, reduction is gain of electrons, you can kind of remember what's what. But what's happening here is that you have 2 reactions or 2 molecules involved specifically from reactions, and when one goes through a reaction, it loses an electron, but then there's one there to gain that electron. And this occurs a lot during cellular respiration, which we'll talk about, how there are certain electron carriers. An example is NAD, and NAD gains electron in the form of a hydrogen, and that is the reduction aspect of the process. And so there are these reactions that work together, and they're able to transfer electrons from one molecule to the next, and those are called redox reactions. Okay, so you'll see some examples of that coming up. Now, another thing I want to talk about here are enzymes. And enzymes are really important aspect of biology, and that's because reactions take place kind of in a slow rate sometimes. They're not necessarily very efficient processes, but enzymes are specific kinds of proteins that can help reactions to take place much more quickly. And so what you would know about enzymes, if you look at the definition, is that they speed up a reaction without them themselves being changed by that reaction. And so when you think about a chemical reaction, you're thinking that there is a product that results from a reactant. So you have some early situation and it changes and makes some later situation called the product. Okay. So for example, with a reaction, you can have, some ways you'll see this A plus B. So these could be two different things. And if they come together and form C, one final product, maybe that's a combination of these two, or let's call it AB. Okay, so these are now joined together. This would be an example of an anabolic reaction that we're just learning about. These are separate, now they're together, joined. Okay. And it can go the opposite way. Let's say you had started out with AB and it became separated. That would be an example of catabolic reaction. Now, this reaction is, let's say it takes 2 days to occur, just randomly on its own. Enzymes are there to make this happen quicker. And so the enzyme, let's just call it E, would interact with the AB in order to help it to make this product. Okay, so let's say this is reactant and the arrow is pointing this way. So this is reactant and this is product. So wherever the arrow points towards, that's where your products are. But in order for this to break apart or be catabolized and becomes two separate pieces, you need an enzyme to help it, okay? So there are lots of different types of enzymes. You'll learn about different ones as we go throughout the semester, but they're there to help different types of reactions occur more quickly. Now, enzyme and substrate. So the enzyme will interact with something in order to help it to become something else, the product. This is called the substrate. Okay, whatever the enzyme is working along with is called a substrate. And what this can look like is that your enzyme could be, let's say, some sort of molecule like this, and your substrate, which is that AB, this, would come here and sit in the enzyme. Okay, so once it's sitting in the enzyme will help to break this apart, so that you can end up with your different separated AB pieces, okay? So enzyme is not going to become a part of the product. It was just there to help it to occur more quickly. All right, enzymes have different places or parts to them that can help them to work better. And one part that is important that you should know about is the active site. So the active site is found right here, this area. And the active site, this is the part where the enzyme is interacting with the substrate. Okay? But your enzyme can be regulated by other molecules. And this is important because if your enzyme is not needed at a moment, you don't want it necessarily to be active because it might start a reaction too early. So you want to have like the key to turn it on. So on another part of the enzyme, you have what's called the allosteric site. And this is where you have a binding site for something else. And this thing will help this active site to become open or available so it can bind to the substrate. But when this allosteric site is not binding its molecule, okay, what happens here is that this active site is not going to be able to necessarily bind this. Okay, so now it's too small. It can't really bind this. But once you have the allosteric site bind to its molecule that activates it, the active site will open up and allow this to come and bind. Really important thing to know is that your active site is very specific for the kind of substrate that it will interact with, okay? So not every type of molecule will be activated or use any kind of enzyme. So specific enzymes work with specific substrates. So that those are specific teammates, so to speak, for those reactions to take place. Another thing that could happen is something called feedback inhibition. And so let's say that we have a reaction that is making a certain product. Let's say that you're making bread, okay? So let's say you have a chef or a baker in the kitchen. They're making bread and sending it out into the dining room. They make bread, send it into the dining room. The baker would represent kind of the enzyme, and the bread would be, the product. So they're making the bread, sending it out, making the bread, sending it out. Let's say, dinner is over and there's nobody else to eat bread anymore. You would say to yourself, it's probably time to stop making the bread, okay? So there needs to be something to stop the enzyme or stop the baker from making so much bread or making too much bread. And so when there's enough bread out there in the dining room, somebody will bring the bread back or bring an empty plate or something back to the baker and say, look, we have enough bread. Please don't make any more bread. Okay? And so this is how feedback inhibition works. Once you have enough of a product, a lot of times it's the product itself that comes back and interacts with the enzyme and shuts it off. So inhibition means to stop something from working or to turn it off. So let's say that if there's enough of that, many times it will bind to the allosteric site, okay? And that will shut the enzyme off, stop it from working anymore, okay? And then usually how the enzyme gets turned on again is that this inhibitor goes away and the activator comes, and then the enzyme is open, the active site is active again, it could work again. So it goes between being active and inactive, depending on what the cell and the body needs. All right? Okay. Now, when it comes to irreversible inhibition, this is something that is scary for the most part, and that's because this can happen with certain poisons and sometimes certain drugs, but hopefully not the certain drugs. But what irreversible inhibition is basically that if something binds to an enzyme or binds to another molecule, and you're not able to remove that thing to activate it again in the future, then you'll never be able to use that enzyme. So let's say that, you told the baker enzyme, stop baking, right? But if, and please this is my example, but if you said stop baking, you're the worst baker ever in history, that might hurt the baker's feelings so much that they will never want to bake again, okay? But we all know that we'll need bread tomorrow and the next day and the next day. So you need to have the baker active again, but not for the whole night. And so that's what happens with molecules. If you have some sort of chemical that binds to the molecule and it shuts it off forever, that is known as irreversible inhibition. It will never get turned on again. And this is a problem in biology because you usually need to have times where you're active as a molecule and times where you're inactive. So irreversible inhibition is kind of permanent, and no longer will that molecule get activated again, and it won't be able to do its job again. So that's why certain poisons, as I said, is a big deal, because they bind to some proteins in your body and they stop those proteins from working at all in the future. And when you don't have normal function in proteins and molecules, your body can't perform certain tasks and duties. So that's really important for you to understand for how to take care of ourselves. All right, so that's it for this chapter.